we have come so far in diabetes research. Just really in the last 50 years has been exponential. So I think it's important to review the history. It's like reflecting on memories to see who you've become as a person, like looking through old photos or remembering those life-changing moments. It's really significant to look at these failures and discoveries because they've really all brought us here today. It's simply amazing to see how science has moved forward and gets hopefully you excited about moving it forward even more. And it's fun, and it makes me personally feel very grateful for the research that has been done. Breakthroughs in research can be small and large, and I love this cartoon here because that's a reaction that you may imagine seeing, but they can be large and small, and they can often be much more emotional than walking through a spider web, but we walk through many webs to get to them, no matter how big or small they are. So let's take a look back at how far we've come. So in the beginning, this was 1550 BC. It's been seen in Egyptian manuscripts describing a disease that caused rapid weight loss and frequent urination. So this was observational. This was an observational trial you could think about. We move forward, they continued to notice a sweet urine. It used to attract ants, actually, and flies, which is how they knew that. And it continued to be monitored for really hundreds of years. It wasn't until the late 1800s that people started to look for a cause of this disease. So the science moved from observing to, what is this? What is going on? During the late 1800s, it was proposed about that mumps was the cause. It was long believed that type 1 diabetes, or diabetes in general at that point, was caused by a virus. And then we had a little bit more of an idea when Paul Langerhans, which you may have heard the term Langerhans, in 1869, got a better picture of what was going on. He was only 22 years old as a medical student, and he made the first detailed description of the microscopic structure of the pancreas. He identified 9 cell types that formed together and rightfully named them. They were named the islets of Langerhans, his name. It was then found, more looking at cause, and about 20 years after that image of the islets, the pancreas was seen, that when the pancreas was removed from dogs, it led to diabetes. So they didn't have the term diabetes then, but it led to them having this sweet urine and what had been described previously. Fast forward to the 1900s, and things started to change. People were seeing it more, and the focus of that research became on managing the disease. So urine testing was done throughout until really the 1960s. In the early 1900s, they would check the urine, and the secretions from those islets that Paul Langerhans had discovered were recognized, and they were named insulin. And they were named insulin because in Latin, insulin actually insula is the Latin term, which means island. And if you look closely at the islets, they look like little islands clusters of cells. Before insulin, until 1921, when it was discovered, they would restrict food and calories, and diagnosis at that point was terminal. So in 1921, we had Frederick Banting. You may have heard the name Frederick Banting, and Charles Best. They teamed together after Frederick Banting had an idea. Actually, he woke up at I believe I was reading he woke up in the middle of the night on Halloween night with this idea. He was on an assignment to do a project on the pancreas and had been reading about experiments on dogs. And he thought, how can I isolate the islets without destroying the pancreas to extract the insulin? And so he teamed up. He went to University of Toronto and was given a lab and teamed up with Charles Best, who was a student there, med student there. And together they ligated the pancreatic duct in a dog and were able to successfully do exactly as he had woken up with the idea. They were able to isolate the islets and establish that the deficiency of insulin was the cause of diabetes. So once they were able to isolate those islets and extract the insulin, they injected it into a 14-year-old to find it to be successful in bringing his blood sugar down. Again, then at that point it was seen in urine, but they were able to do that. Eli Lilly started making quantities of insulin from cows because there weren't enough dogs to create enough insulin for those who needed it. Frederick Banting opened up a hospital to treat people living with diabetes. And so you can see that this breakthrough really projectiled into treatment, different treatment than had been done. They also noticed in the early 1900s that there was a seasonal onset. So there were fewer cases in May and June than in the spring and the fall. And at that point, they still correlated that to a viral trigger because it coincided with outbreaks of mumps. So there were additional changes as we went along in insulin, making insulin longer. This was all done through clinical research. As we go along through the 1950s and 1960s, the research was really focused on home management, and we learned about autoimmunity. So you may have heard autoimmune when you were diagnosed, or I hope you understand that type 1 diabetes, as we know, is autoimmune. And so that was really only discovered in the 1950s. It hasn't even been 100 years since that's been known. When it was discovered that autoimmunity is even a thing, it really started a paradigm shift in research and in the medical field. So when it was first, the term autoimmune was brought about, diabetes was not in that realm. They didn't include diabetes as autoimmune yet. Insulin discoveries and changes in how they made insulin continued to change, so management got better. You can see that there was a first insulin pump in the late 1960s, like it was really 1970s. And it was, can you imagine wearing the big old insulin pump? They've come a long way thanks to clinical research. So we've come a long way. As we got into the 1970s, we started to correlate genetics. Researchers were seeing a correlation between family members. We also continued to get better insulin. So in 1973, there was a big step in that thyroiditis. So it wasn't diabetes. You can see how all the research overlays each other, especially when it comes to autoimmune disorders. We know they're related. It was shown that immune response genes could make people more or less susceptible to viral infection triggers for autoimmunity. In the same time period, researchers were looking at HLA, which is part of genetic coding, genetics, and found an association with other autoimmune disorders, still not diabetes in there. It wasn't there yet, but the association with lupus and MS, and so we were seeing a connection between autoimmune disorders. And then in a little bit soon after, we found type 1 diabetes to have what are called autoantibodies. And we'll get into those a little more. But that gave us and people working in diabetes research a recognition that type 1 diabetes was an organ-specific autoimmune disease, so specific to the pancreas. Alongside all of this, as people are being treated more for diabetes, insulin was improving as it went along. Now we get into a little closer to where we are. So in the 80s to the 2000s, we had better glucometers. We were moving away from checking urine. It became a standard of care for people to check their blood sugar at home. We, in 1999, had the first professional CGM, which would keep people on it for providers to watch for trends. And then in 1993, which was not that long ago, I feel like it wasn't that long ago, one of the largest studies called the Diabetes Control and Complications Trial, or it's also termed the DCCT, was a huge 10-year trial with over 1,400 people, clinical trial participants, showed that good glycemic control, so keeping blood sugar in a target range or a hemoglobin A1C within a target, the data was able to show that it prevented up to 70% of macrovascular complications. So that was a piece of data that we now had to show if we can manage it and keep it in range, then we can prevent long-term damage. Now you see that something like the DCCT, again, this clinical research, all these people that participated really built diabetes management the way it is today. So now we have standards of care and we know what targets we need to work towards to prevent complications. And we have data to show that and evidence to show that. So just in the last 20 years, in the last 40 years, we had connected dots with autoimmunity, genetics, and tools for management. And we continue to move that diabetes research forward in all of those areas. But in the last 20 years, we've really honed in even more. So in 2000, the Surgeon General identified diabetes. Now we had been 7 years out of the DCCT, had identified diabetes as a national health objective, and it triggered much more focus on research. In 2003, there was the end of a 9-year study, the DPT-1, and that was working towards prevention of type 1 diabetes using oral and injected insulin. It was a negative study, so it showed that oral and injected insulin did not prevent the autoimmune response, but that negative study pointed us in another direction. So again, even the negative ones move us forward. And so after that, TrialNet, Type 1 Diabetes TrialNet was created to continue that work in preventing Type 1 diabetes. So other studies continued and still do. We brought us here to 2019, where just this year there was a positive study presented that teplizumab, which is also called anti-CD3, was shown to delay onset of type 1 diabetes when it was given to people who were at risk, so antibody positive. And again, I'll go more into the antibodies. And it was shown to have a delayed onset of clinical disease, so of type 1 diabetes, for an average of 2 years. And I'm not going to go into a whole lot of details, but we'll review some of the results of that study as we go along. It was very exciting for the diabetes research community. So now as we go into what kind of research you may be being reached out about, or maybe interested in being part of, or want to know what's going on, there's a number of different